Hello and welcome to part 1 of chapter 15. In this lecture, for this lecture, we're going to talk about DNA mutations, and then in part 2, we'll talk about DNA repair, and we'll touch a little bit on the development of cancer. So in mutations, a mutation is a change in the genetic material, usually in the code, in the actual DNA code. And this can be anywhere from a whole section of a gene could be replaced with an incorrect code, maybe just a couple of bases, even all the way down to a single base being changed or added or deleted. Mutations, however, are essential for continuity of life. And what we mean by that is that mutation is ultimately responsible for evolution of a species over time. Without some kind of mutation, we would have no possible way to change genetic expression in response to environmental changes. So mutations allow for an extensive, almost innumerable number of different variations or alleles of a genes that allow the continuity of an organism or a species in response to changes in the environment. New mutations, for the most part, are usually more harmful than they are beneficial. Evolution of most species has become very advanced, is very advanced. So any new mutations are probably not going to allow the organism to survive in a better way than it already is as far as a species goes. But that does not mean that there are not mutations out there that can occur that either are benign, don't really make any difference at all, or can actually be beneficial to an organism, especially in a changing environment. So gene mutations themselves alter the actual sequence of the DNA itself. This can occur within an actual structural gene. It can occur in a non-structural gene. It can also occur in promoter sequences, operators, enhancers, silencers, anywhere in any portion of the genetic material, these mutations can occur. Now, a point mutation is a mutation that affects a single base pair. Usually it's some type of either addition, deletion, or substitution. The two most basic alterations are, again, changing the base sequence, so changing an A to a C or a G to a T, or an addition or deletion, adding another nucleotide or removing a nucleotide. And in doing this, you throw off the reading frame of the gene itself. If we add a base or remove a base, this is going to change the codon sequence, so it throws off the reading frame. So here are some examples of some changes in a base sequence. So first we have a base substitution. You can see here that we should have our second codon here should read GCT, but because there's a base substitution, the T got changed to a G. This is going to cause its base, its complementary base, to change from a G to a C. So we have this this kind of change here in the base, from an A to a C. And regardless of which strand the gene is on, whether it's running this way or that way, we have a base substitution here that has changed the actual sequence. This should be GCT, and it's now GCG. And in addition or deletion, this is where a single base has been added or removed. And in this case, the addition or deletion of a base is going to throw off the reading frame drastically. Now let's take a look at our reading frame. Here on the left is our normal gene, what we would call the wild type. So our first codon, our second codon, third codon. But if we move over here, an A has been inserted into the middle of this gene sequence. So although our first codon is the same, our next codon is now different, which in turn is going to change every codon thereafter. Normally, the codon should have been GCT, but because this A got added in here, and it's going to have a complementary base, the T will be added there, this again is going to throw off the reading frame, regardless of which direction the gene is running in on the double strand. So I believe our video is not working. So we'll put a link to that in the website. It's not working right now, so we will move on. Now, sometimes the way that these mutations affect the amino acid sequences are kind of interesting. Now, we can have what are called silent or neutral mutations. Remember the codon table. I mean, you don't have to memorize it, but remember on the codon table, we had start codons, we had the codons that encoded for amino acids, and then, of course, we had the stop codon. In a silent or neutral mutation, here, the change in the amino acid sequence, this would only be with the base substitution. This would not occur with an addition or deletion. So this is a base substitution only. If we had a single base that is replaced, a G replaced with a T or an A with a C, if that's the case, sometimes those base substitutions will result in a codon that still encodes for the same amino acid. So in this case, it may not be the wild type DNA sequence, base pair sequence. However, the amino acid sequence of the functional protein that is the product of that gene will be the same. So this is a mutation in the DNA sequence, but because it results in the same amino acid, it's referred to as a silent mutation. Then we have the missense mutation, and in a missense mutation, a single amino acid in the final polypeptide product of the gene is going to be different. This can be a disease state, like sickle cell anemia is a missense mutation. That is the result of a single base substitution, which in turn results for a single amino acid being different in the hemoglobin protein. So missense mutations are just where we just change one amino acid. And of course, again, depending on the amino acid that gets changed, if it's a hydrophobic amino acid being replaced with another hydrophobic amino acid, then that's not going to be such a horrible substitution. It may slightly alter the function of the protein, but it's probably going to keep the actual structure itself fairly similar. So the protein may still function. But if we replace a hydrophilic with a hydrophilic with a hydrophobic, well, then that's a completely different type of structure. Remember, those amino acids are going to first interact with the amino acids surrounding them. So if we have a hydrophilic and a hydrophobic, or a positive versus a negative charge, that can drastically alter the structure of the protein, thereby altering the final function of it. Sickle cell anemia, like I mentioned earlier, is a missense mutation where we have a single base substitution. So we have an A in the second position of the codon got replaced with a T. This causes what should be glutamine to be replaced with valine in the sixth position. In turn, this results in a blood cell where the hemoglobin is not folded properly and the cell itself becomes sickle-shaped instead of discus. A nonsense mutation occurs when we're still looking at point mutations here. A nonsense mutation is when we get a stop codon being encoded for instead of an amino acid. So we're, moving along in our protein here. And here, this C got substituted with a T. But TAG, if we translate that into codon, encodes for a stop codon. This means that the protein is going to be cut short. It'll be incomplete in its structure. It's what they call a truncated polypeptide, and it's going to be non-functional. Finally, we have a frame shift mutation, and this type of point mutation is an addition or a deletion. So again, I showed you in a slide before, but just again, by inserting right here is our CAT. This should be our first codon here, but in a frame shift, everything gets moved over one. Now this could be a deletion, this could be an addition, but we had 200 bases in that gene, and now we have 201 or 199. And again, this is going to throw off not only that first codon, but it's going to throw off every codon thereafter, so that what should have been this amino acid sequence now becomes this amino acid sequence, and it's not even remotely the same. So the protein will not fold properly, and it will be non-functional. Our movies are not working this evening, so we will have to post those. Now, what about mutations outside of the actual gene sequence? There are mutations that occur in the promoter region or in the core promoter. If we have a mutation that occurs in the promoter, we can, this is called an up promoter or down promoter, we can increase or decrease activation of that particular gene. Sometimes there can be the transcriptional response element, or also known as the operator site, which is pretty much right in here. This can alter how the gene is actually regulated, when it's turned on, when it's turned off. It can make it harder to turn it on or easier to turn it on. Then we have splice junctions. So remember, in eukaryotic DNA, we have regions in our actual gene itself, those little regions that are actually introns, and there's part of those introns are the splice sites. Well, if the mutation is within the splice site, then the spliceosomes are not going to recognize it. They will not bind to it properly, and what should have been processed out of that the pre-mRNA during RNA processing will remain behind. So either the splice site is, we have a mutation that results in a splice site not being recognized. You could also have a mutation where a splice site, it just results in what is a splice site where it shouldn't be. Then we have translational response elements. These can prevent the proper regulation of the translation, and this will occur up here in the very front of the promoter. And we can have, it can be more difficult for transcription factors to bind to the core promoter. It can be really difficult for RNA polymerase to get activated as well. Now, is a mutation what we call a germline or a somatic cell? Now, a germline mutation is one that is inherited, and it occurs in the actual gametes themselves. So germline mutations occur in sperm and eggs. Somatic cells are all the other cells in the body, so everything except for gametes. And if we have mutations occur sporadically in a somatic cell, then we can see areas that are more localized in that. An example of that is a genetic mosaic. In a genetic mosaic, this is oftentimes expressed in changes in skin color. And so somebody may have dark patches or light patches on their skin that are very, very distinct. And that is known as a somatic cell mutation. So what are some of the different causes of genetic mutations? These tables are in your book, but just to look at them, sometimes there's errors during DNA replication. Maybe DNA polymerase III added the wrong base and it didn't get caught during proofreading. Sometimes there are toxic products from metabolism, such as reactive oxygen species, hydrogen peroxide, superoxide dismutase. These are natural of what we sometimes call free radicals that we produce in our own cells, and if they're not neutralized quickly, they can actually react with, get into the nucleus, react with DNA, and cause mutations. Sometimes there are spontaneous changes in the actual structure of a nucleotide. How the hydrogen bonds, maybe they're not forming quite properly, or during translation or transcription, that particular a base got altered somehow in its actual structure. UV light can do that as well. Then we have transposons. Now, in transposons, we haven't really covered yet, but transposons carry out a process known as transposition, and transposons are pieces of DNA that jump from one region of a chromosome to another, or they can jump from one chromosome to another. So they're very similar to insertions and deletions. Then we have induced mutations, right? So up here, these are spontaneous mutations. Then we have induced mutations, and these are things like carcinogens and mutagens, chemicals, physical agents, things that interact with the DNA that we get exposed to. So our next lecture in part 2, we're going to go ahead and end here for part 1, and in part 2, we're going to talk about the different mechanisms of DNA repair. See you on the flip side.